In 2000, Melanie Culver and colleagues at the Laboratory of Genomic Diversity in Frederick, Maryland, published a genetic analysis of pumas collected from across North and South America, one of the most comprehensive surveys of a large felid's population genetics attempted up to that point, using microsatellite markers and mitochondrial DNA sequences from animals representing populations across the entire species range. The results were unexpected in a specific and significant way. The pumas of North America, sampled from populations spanning Canada, the western United States, Central America, and down into the northern parts of South America, showed levels of genetic diversity so low that the entire continental population appeared to descend from a very small founding group. The microsatellite markers and mitochondrial sequences used in the analysis told a consistent story. At some point in the relatively recent past, the North American puma population had passed through a severe genetic bottleneck, a catastrophic reduction to a tiny number of individuals, and had expanded from that remnant. The timing suggested by the molecular clock estimates placed this bottleneck at the end of the Pleistocene, approximately 10,000 to 12,000 years ago. This was not a coincidence. It was the same period in which North America's Pleistocene megafauna, mammoths, mastodons, horses, camels, ground sloths, and a suite of large predators went extinct across the continent. The original North American pumas, the ones that had coexisted with those animals for hundreds of thousands of years, had gone extinct along with them. The pumas living in North America today are not their descendants. They are descendants of a South American population that expanded northward after the original inhabitants were gone, recolonizing an empty ecological niche on a continent their ancestors had left behind. Puma concolor is, by most measures, the most ecologically successful large felid in the Americas. It currently ranges from the Yukon in northern Canada to the southern tip of Patagonia, a north-south distribution of roughly 12,000 kilometers that makes it the land mammal with the widest latitudinal range in the Western Hemisphere after humans. It occupies habitats ranging from tropical rainforest to alpine tundra to coastal desert to suburban edge habitat where it preys on deer in the hills above major cities. It preys on animals from white-tailed deer and elk to small rodents and even insects in lean times, adapting its diet with a flexibility that most large predators cannot match. A puma in the Yukon hunts moose and caribou in boreal forest. A puma in coastal California hunts mule deer on chaparral hillsides 2 kilometers from the Pacific Ocean. A puma in the Patagonian steppe hunts guanacos across open grassland. It is the same species doing all of these things, and the genomic work of the past 2 decades has begun to reveal how much of that flexibility is baked into the puma genome and how much is learned behavioral plasticity on top of a generalist genetic substrate. It has more common names in English than any other animal: cougar, mountain lion, catamount, panther, puma, a testament to how many different human cultures have encountered it across its enormous range and given it a name independently. Despite all of this, the puma is not a big cat. The family Felidae is divided into 2 major subfamilies: Pantherinae, which contains the roaring cats, lions, tigers, leopards, jaguars, and snow leopards, and Felinae, which contains everything else, including domestic cats, cheetahs, lynxes, ocelots, and the puma. The puma is a member of Felinae. It cannot roar. It communicates instead with a range of sounds, including whistles, chirps, and the eerie screaming call that has made it the subject of more campfire stories than perhaps any other North American predator. The question of which living cat species is most closely related to the puma was settled by genomic data in the early 2000s and confirmed by progressively more complete whole genome studies in subsequent years. The puma's closest living relative is the cheetah, Acinonyx jubatus, the fastest land animal on Earth, native to Africa, and small remnant populations in Iran. The two species diverged from a common ancestor approximately 6.7 million years ago, according to molecular clock estimates from the 2006 Johnson et al. Felid phylogenomics study published in Science. The next closest living relative is the jaguarundi, Herpylurus jaguarundi, a small, dark, elongated felid found in Central and South America that looks superficially more like a weasel or an otter than a cat. All three species, puma, cheetah, jaguarundi, form a clade called Acinonacini, whose common ancestor diverged from the lineages that would produce lions, leopards, and the other big cats well before either reached its current geographic range. The physical similarity that most people perceive between a puma and a lion, both are large, plain-coated, powerfully built felids, is not a close relationship. It is a superficial resemblance between two animals that parted evolutionary company millions of years before either achieved its modern form. The cheetah connection becomes less surprising when you examine what pumas and cheetahs share beyond their genetic relationship. Both are lean, long-legged felids built for speed and pursuit rather than the ambush and grapple strategy of most large cats. Both lack the ability to fully retract their claws, a trait that is otherwise nearly universal in felids and that in pumas and cheetahs reflects a shared evolutionary heritage of using claw traction for high-speed running rather than gripping prey during an ambush. Both have proportionally small heads relative to their body size and relatively small teeth for an animal of their mass, a configuration suited to chasing down prey and delivering a suffocating throat bite rather than subduing large, struggling animals with brute force. These shared traits are not convergences that evolved independently in two separate lineages. They are retentions from a common ancestor, features of the ancestral Acinonacini body plan that both the puma and the cheetah lineages have preserved in different environments on different continents. The cheetah took this ancestral running specialist template and refined it for extreme speed on open African savanna. The puma took the same template and generalized it for a wider range of environments and prey sizes, becoming one of the most adaptable predators on Earth rather than the most specialized. In the Pleistocene fossil record of North America, the puma had a close relative that pushed the pursuit predator specialization further than the puma ever did. Maracinonyx is a genus of extinct North American cats that appears in the fossil record from approximately 3.2 million to 10,000 years ago. Two species are recognized: Maracinonyx trumani, which appears to have been a genuine high-speed pursuit predator with limb proportions very similar to the modern cheetah, and Maracinonyx inexpectatus, which was more puma-like in its proportions and may have been more of a generalist. The relationship between Maracinonyx and living cats has been debated since the genus was first described in the 1970s. Some researchers classified Maracinonyx as an early cheetah that had crossed into North America from the Old World. Others placed it as a close relative of the puma that had independently evolved cheetah-like features on the same continent. Ancient DNA analysis, using material extracted from Pleistocene fossil specimens, has supported the second interpretation, placing Maracinonyx within or very close to the puma lineage rather than within the cheetah lineage. If this placement is correct, North America in the Pleistocene had its own cheetah-like pursuit predator that was not a cheetah, but a relative of the puma. And the prey species of North America, pronghorn antelope in particular, which remains the fastest land animal in the Western Hemisphere today, and whose speed far exceeds any living predator it currently faces, may have evolved their speed in response to predation pressure from Maracinonyx rather than from any living threat. The Pleistocene extinction event that eliminated Miracinonyx also eliminated the original North American puma populations, the American horse, the American camel, the woolly mammoth, the mastodon, the giant ground sloth, the American lion, the saber-toothed cat Smilodon, and the short-faced bear, the most comprehensive loss of large mammal diversity on any continent outside of Australia in the last 50,000 years. The continent went, in the space of a few thousand years, from supporting more large mammal diversity than modern Africa to the ecologically impoverished state familiar to us today, where the bison and the white-tailed deer are the largest herbivores, and the wolf and the grizzly are the largest terrestrial predators. The cause of this extinction event remains one of the most actively debated questions in quaternary science. Whether human hunting pressure from the first Americans arriving at the continent, climate change at the end of the last glacial maximum, or some combination of the two was the primary driver. For the puma, the genetic record provides a clear timestamp on the catastrophe regardless of its cause. The bottleneck in North American puma diversity dates to the boundary of the Pleistocene and Holocene, approximately 10,000 to 12,000 years ago, consistent with the timing of the broader megafauna collapse. Whatever killed the mammoths and the horses also killed the original North American pumas. The South American puma populations, which did not experience the same scale of extinction pressure at the same time, South America lost some megafauna but retained more large mammals than North America, retained substantially greater genetic diversity, and it is from those populations that the recolonization of the empty continent proceeded as ecological communities reorganized in the early Holocene. The recolonization of North America by South American pumas was not a single event with a known date. It was a gradual northward expansion by animals following prey into newly available habitat as Pleistocene conditions gave way to Holocene ecosystems. The genomic data does not allow a precise reconstruction of the route or timing of the expansion. The fossil record of pumas in North America during the early Holocene is sparse, and radiocarbon dates on puma remains from the period immediately following the megafauna extinction are few. But the pattern of genetic diversity across living North American populations is consistent with a south to north founding event, followed by gradual range expansion as prey populations and habitats stabilized. Populations in Central America and the southern United States retain somewhat more genetic diversity than those further north, consistent with geographic proximity to the source population. The 2019 study by Sarami and colleagues, which used whole genome sequencing rather than microsatellite markers to analyze puma population structure in unprecedented detail, confirmed the low heterozygosity of North American pumas relative to South American populations and identified signatures of population expansion consistent with a recent recolonization. The genetic consequences of the bottleneck are still visible in the genome of every North American puma alive today: reduced diversity at immune-related loci, elevated frequency of slightly deleterious alleles that would normally be purged by selection in a larger population, and genomic signatures of inbreeding that, while not severe enough to compromise the fitness of individual animals in most populations, have left the species more vulnerable to novel pathogens and environmental stressors than a population with full genetic diversity would be. The Florida panther is the most extreme case of the genetic impoverishment that isolation and small population size can produce in a puma population. The Florida panther is the last surviving puma subspecies east of the Mississippi River, a remnant population that, by the 1990s, had declined to fewer than 30 individuals confined to the southern tip of the Florida Peninsula. At that population size, with no immigration from other puma populations and no possibility of natural dispersal past the barriers of human development that had fragmented the landscape, inbreeding had become severe. The genetic consequences were visible in the animals themselves. Florida panthers showed a high incidence of kinked tails, a developmental abnormality caused by inbreeding, as well as heart defects, undescended testicles in males, and poor sperm quality. Immune function was compromised. Mortality from disease was elevated. The population was not just small, it was in active genetic deterioration, accumulating inbreeding depression faster than natural selection could remove it. The consequences of passing through a genetic bottleneck, visible only in statistical patterns in the genomic data for North American pumas generally, were playing out in real time in Florida in the bodies of individual animals. The response to the Florida panther crisis produced one of the clearest demonstrations in conservation biology of what genetic diversity does for a population. In 1995, wildlife managers introduced 8 female pumas from a Texas population into the Florida Panthers range in southern Florida. The decision was controversial at the time. Purists argued that introducing animals from outside Florida would dilute the genetic distinctiveness of what had been classified as a separate subspecies, Puma concolor corii. Geneticists argued that the Florida panther was already so inbred that the concept of subspecies distinctiveness had become biologically meaningless. A population deteriorating toward extinction is not a genetic resource to be preserved. It is a demographic emergency to be addressed. The geneticists prevailed. Texas pumas are genetically similar to Florida panthers. Both descended from the same South American recolonization of North America, but had been isolated from Florida for long enough to have accumulated different alleles at many loci. The introduction was, in genetic terms, a controlled transfusion. New alleles flowing into a genetically depleted population at a calibrated rate. Within 2 generations, the results were measurable and striking. The incidence of kinked tails in the population dropped by more than half. Heart defects became substantially rarer. Sperm quality improved to levels comparable to genetically healthy puma populations elsewhere. Kitten survival rates increased significantly. The population grew. By the early 2000s, Florida panther numbers had more than doubled from their pre-rescue low, and the improvement in fitness traits was directly and causally attributable to the genetic input from the Texas animals, rather than to any change in habitat or prey availability. The Florida panther rescue became a landmark case study in genetic rescue and demonstrated conclusively that the loss of genetic diversity has direct, visible, measurable consequences for real animals, and that those consequences can be reversed. The Puma genome, published by Seremi and colleagues in 2019, provided the most comprehensive molecular portrait yet of what a population looks like after passing through a near-extinction event and recovering. The genome showed that North American pumas carry an elevated burden of homozygous deleterious variants, positions in the genome where both copies of a gene carry the same slightly harmful allele, a signature of inbreeding that allows recessive effects to be expressed. It showed reduced diversity in immune gene families, particularly the major histocompatibility complex genes that govern the recognition of pathogens, precisely the gene regions where diversity is most important for a population's ability to respond to novel diseases. And it showed the signature of a population expansion following the bottleneck, a sudden increase in effective population size in the relatively recent past, consistent with the recolonization history reconstructed from population genetic data. The genome also revealed something about the puma's resilience. Despite carrying these genomic scars, North American pumas have managed to maintain large, widely distributed, ecologically functional populations across most of their historical range. The species has survived the loss of the Pleistocene megafauna that it evolved alongside, the extinction of its original North American populations, a recolonization from a small founding group, and centuries of human hunting and habitat modification. It is carrying a compromised genome by the standards of what a large felid's genome should look like, and it has thrived anyway. Pull all of this together, and the puma that emerges from the genomic record is an animal whose history is almost entirely invisible from its biology alone. Standing in front of a puma, you would have no indication that you were looking at an animal whose closest relative lives on the African savanna, whose North American lineage went extinct 10,000 years ago, whose current North American population descends from a tiny founding group from South America, or whose Florida subspecies was in active genetic collapse within living memory and was rescued by a deliberate human intervention. The puma looks like it belongs to North America, to the mountains and forests it currently occupies, to the ecological role of apex predator it fills. The genome says something more complicated, that belonging, in this case, is approximately 10,000 years old, that the puma is a replacement for an animal that was genetically distinct from it and that went extinct alongside the continent's horses and elephants, that the ease with which the puma filled the vacant niche, spreading from South America, across Central America, and up through North America in the Holocene, says something important about how resilient large predators can be when they have the right basic toolkit and an open ecological stage. And that the genomic damage visible in every North American puma alive today is the record of how close the species came to losing even that opportunity, and how much of what we take for granted in a living population depends on diversity that is invisible until it is gone. Subscribe. New discoveries about lost species posted every few days.